Hello, 1604.
We're here.
Okay, so you hear and see me?
In a pit, but yes.
Yeah. If I change it out of the pip, then it gets all messed up. So
Okay.
start off in the pip. The system is a bit strange down here. I can't zoom the camera in or anything like that. We'll just go with it like it is today and get to the lecture. I have a meeting here at Brooks just after this lecture, and then I have to go over to Northwest Vista for a meeting there. So I'm lecturing from the Brooks facility today. So we begin today with Lecture 27, the first lecture on the cardiovascular system. And I remind you that the second lecture, 28, I've split it into two, 28.1 and 28.2. And then Lecture 29 are being pre-recorded, and they will be posted as streaming video. So as we had stated, for this Friday, and, Marco, are you doing the one next Friday?
Yes, sir, I am.
Yeah. So there will be an in-lecture class Friday, what's that, the 14th, and the Wednesday before that. But this Friday, I have a seminar at St. Philip's College, so I'm posting the pre-recorded lecture. And then on next Monday, I'm at a meeting, the neuroscience meeting in New Orleans. So the Lecture 29 will be pre-recorded and posted for that. So we begin with an overview. Remind ourselves that the cardiovascular system is the first major system to develop, week 3. So we've seen this already. We've introduced the cardiovascular system, as we said, as we're proceeding through everything. Now, of course, we begin to focus on the organogenesis of the cardiovascular system. So things happen pretty fast. It's functioning and circulating blood by week 4. And there is a diffusion across the placental membrane that is inadequate without movement of blood. And we've made this point repeatedly throughout the semester. So we recall that the splanchnic mesoderm was surrounding the embryo, and it was contributing to the formation of the heart that we saw in front of the prochordal plate. We will also see that the paraxial lateral plate mesoderms and some neural crest cells kick in. The neural crest cells, of course, are going to be associated with development of vasculature as the neural crest mesenchyme within the arches. Early angiogenesis formed the generalized blood vessels, and we've discussed that already, so we won't repeat or have any focus on that. Primordial heart is evident at 18 days and can be beating as early as 22 days, somewhere around there. So the paired angioblastic cords are what we're going to look at first, just in front of the prochordal plate, as we recall. That was the cardiogenic area that we saw in the 3-week embryo. The cords are going to develop a lumen and form endocardial heart tubes. So it starts as paired tubes that are going to fuse together, as we will see. This slide is a dorsal view of the embryo. And we're going to look at a transverse section and a mid-sagittal section to focus our attention on the developing cardiogenic area here just in front of the prochordal plate. And of course, the cut edge of the amniotic sac is in white here, and that reminds us of that reference point that we've used from the very beginning. So we recognize this sagittal view, the mesoderm that had infiltrated out into these areas, the extraembryonic mesoderm. The green in this case is showing the endoderm lining the yolk sac, and the surface ectoderm is shown in blue. So slightly different colors than what we used before, but no harm. The red coloration is that angiogenic cluster that we had talked about. Starts off as several clusters, extending from caudal to rostral in front of the prochordal plate. A cross-section through this area shows us that it reminds us that it is a horseshoe-shaped structure, as we recall. Well, the pericardioperitoneal canal is beginning to form. We see the extracoelomic spaces forming out there. So we recall all of those things. So the pericardium, pericardial sac is beginning to form just dorsal to these angiogenic clusters. So they are on either side, and again associated with that horseshoe-shaped pericardioperitoneal canal. You also recall that the rostral, the head fold, tail fold is occurring as well, but the head fold and the lateral folds were extremely important for moving the heart into the chest and bringing it together to fuse in the midline. So we're looking at day 21 in the upper panel and day 22 in the lower panel. Now we have the red showing the mesoderm, and we see this fine black line showing the endocardial tubes. The dorsal aorta is shown here, and the notochord as well. So we recall with the lateral folding, we get the progression toward midline, ventral midline for the fusion. At the same time, this prochordal plate is coming around to form the oropharyngeal membrane. And the two tubes we see will fuse together, the endocardial tubes. They'll have a dorsal mesocardium that eventually breaks down. So this is nothing new to us. This is just introducing those ideas to us again. So now we'll kind of back up and review what's all of these events that are happening. So the figure 14.9 from our textbook shows us the heart tube, or the endocardial tube, and the pericardial cavity forming just rostral to the prochordal plate or the future oropharyngeal membrane. So we're being shown the arrow for the head fold to bring that in. So the heart tube starts off rostral to the foregut and to the rostral neural folds. They're closing. And then eventually will become ventral to the foregut and caudal to the developing forebrain, as we see here. So we recall this. When this head fold is occurring, at the same time the lateral folds are occurring. So the two tubes are being drawn in and together, and they fuse in the midline, ultimately to form a single tube, the embryonic heart. So the septum transversum is right nearby, so there's a lot of vessels developing in this area, in the mesenchyme of the septum transversum, and as well in the mesoderm and all the surrounding areas. So this is the lateral view of the day 18 to 21 embryo, human embryo, taken from Gray's Anatomy. We focus on the yolk stalk and the ventral aspect, the body stalk a little bit caudal to that, and the cut edge of the amnion. We recognize the somites, developing nervous system back there, and then this, of course, is the gut tube bulging on the surface. And we see the heart bulge here, as we had seen before, and the visceral arches. So we can envision blood flow coming through the heart. This way, it's now a single tube, then it's going to split, and there will be arches, the arteries, the aortic arches, or pharyngeal arches. So what is the location of the cardiogenic area in the pre-somite embryo? So it's in that splanchnic mesoderm in front of the neural plate, prochordal plate as well, on both sides of the embryo. That's very important because has established its connections with the dorsal aortas. And as we said, the heart will begin to beat on around day 22. So the tubes will fuse in the lateral plane to form the primordial heart, as we had mentioned. We'll see that in figures 14.7 and 14.8. It beats during week 4, as we indicated, so there will be blood flowing at that time. The formation is induced by the underlying endoderm. So the beginning of the formation of the foregut is a signal for the final development, or for the initiation of the final developmental processes to produce the adult four-chamber heart as we know it. So this is figure 14.7, and we are looking from a ventral view of the heart, so from the front, and the pericardium has been cut open. So we see the cut edge of the pericardium and the pericardial cavity, of course, and then we see the two heart tubes, let's just use that term. And we know the heart has 3 layers. The epicardium isn't quite formed yet, but it will be on the surface of this structure. And then the myocardium is the middle layer, the muscle layer. And then the endothelial lining will form the endocardium. At this time, the tube is filled with a cardiac jelly, so we haven't quite opened it up yet to get the full flow of the blood. And we see the rostral continuation is through the first aortic or pharyngeal arch. That arch is just rostral and a bit lateral to the foregut. And we see on the venous end, the caudal end, we just see quickly the vitelline vein and cardinal veins draining into this structure. So now in panel B, we're seeing the beginning of the fusion of the two heart tubes together in the midline. And we see the thick myocardium. So the number of cardiac myocytes is increasing very rapidly. And they fuse together at the rostral end and maintain their communication with the first aortic arch. And then here in panel C, we see that the fusion is complete. We can identify early areas. So the endocardial heart tubes are shown as they meet in the midline. And so this is where the primordial atrium is going to be, because remember that the venous return is coming from the caudal end in. And then the primordial ventricle is present, and then the bulbous cordus. We'll see that this develops into basically the top of the ventricles as they feed into the major vessels that exit the heart. At this stage, we have 2 venous returns into this common tubular chamber and the 2 arterial exits at the rostral end. A lot of things are happening in here from this stage on that are going to direct the flow of blood and eventually split the chamber, single tubular chamber, into the 4 chambers as we know them. It's not the focus of this lecture. Right now we're just doing the overview of the development, and then lecture 29 is on the development of the heart. So this large vein coming in at the caudal end is the umbilical vein, coming right straight into the heart on the venous end, so it would be bringing in the nutrient-rich and oxygenated blood, and it would be joined by the systemic venous drainage, deoxygenated and nutrient-poor, let's say, blood. So there is a mixing of the blood as soon as it hits the heart in the embryo. We begin to see that there's just a little bit of a twist that's beginning to appear. It separates, just delineates, let's say. There's no internal separation yet, but delineates very nicely the ventricle from the bulbous cordus. So which of the following is the correct sequence of structures of the developing heart? So the sinus venosus leads into the atrium and then to the ventricle, and of course, what isn't shown here, but still this is the correct list. So lists don't have to be complete, they just have to be in the correct sequence. The bulbus cordis would be on the end of this. So now we see at this stage in panel E, the umbilical vein draining in again. And on the opposite side, we see the vitelline vein and the common cardinal vein. So these are the primary venous structures that are present at this time, giving blood flow into the atrial end of the heart. So we see the primordial atrium now is even pushed a little bit. Remember, this is a ventral view. You're looking from the front. So the atrial part is starting to get pushed to the back, and we'll see that it even gets pushed up rostrally. So the atrial end, the original caudal end of this single heart tube, is going to be displaced first dorsally and then rostrally to take its adult position up surrounding the structures that the truncus arteriosus becomes. So we begin to see that the ventricles are even delineated by these slight indentations. So there's a lot of changes that are occurring in the walls of the heart as that tube is elongated. It can't fit anymore, so it's kind of getting contorted and goes through a bit of a rotation. And it's not really getting twisted and rotated, but growing and developing and just gets displaced from the mass of the myocardium that is developing. So we see the future right ventricle really is feeding right now into the truncus arteriosus, but the future left ventricle is also sharing that common canal going into there. And we're still maintaining this split into the first aortic arch. We see the second aortic arch there. So a lot of changes occurring in these vessels on the arterial and the venous ends, as we just saw here, and that is part of Lecture 28. So we won't focus on that anymore at this time. So just an overview on this developmental process that is occurring. We recall that the early heart consists of the three layers: endothelium, the inner lining, a single cell layer thick of flat cells, so it's a simple squamous epithelium. The cardiac jelly filling the chamber, and the myocardium or epicardium, and then the epicardium will be added to it a bit later. Remember, the heart, we talked about it as being a balloon, an organ like our hand that grows into the balloon, the pericardial sac that is developing. So the surface layer, the epicardium, will be added just a little bit later. For right now, it's just a lot of mesenchyme in that area undergoing tremendous division and differentiation. So because of that cranial folding, the heart lies ventral to the foregut and caudal to the oropharyngeal membrane, as we had mentioned. So now we can follow through with figure 14.8 and see how the lateral folds bring these two together through looking at cross-sectional views. So panel A is about day 20, and then panel A is a transverse section at about this level. We recognize in this dorsal view of the embryo, so we now are at the 3-somite stage. So we've already had the beginning of the movement ventrally and caudally. So the pericardial coelom, or the pericardial peritoneal canals, are forming in this area. So that's our cue that the heart is forming right nearby. So it is then pulled down, and now it's going to begin to be pushed together. And we see through this plane of section here, as we move from the surface ectoderm deep, we will encounter the pericardial coelom first. And then as we go deeper, we will see the endocardial heart tube, and there is one on each side, left and right. So we see the endoderm is still nearby as the foregut was formed. So this is triggering these mesenchymal cells in this area to begin their differentiation as cardiac myocytes, not as the smooth muscle that lines the other vascular structures, where we would find the smooth muscle. Now we see here in panel C, so that was day 20 in panel A and B. Then in panel C and D, it's going to be a couple days later, and then panel E is going to be 28 days later. Excuse me, day 28, about 8 days later. So in the matter of a week, these cardiac tubes have met in the midline. The mesenchyme is kind of meshed all together. We see that in a sense, the heart is beginning to push into the balloon, the pericardial sac, but there's still a lot of mesenchyme surrounding the heart tube. So this mesenchyme is going to be differentiating into the myocardium and then thinning out ultimately. So we see the cardiac jelly out in this area here. The epicardium now has formed by day 28, and of course the endothelial lining is present. And the myocardium is just on the surface. And now in panel E, we see we have the linear single tube with the two umbilical veins feeding into the sinus venosus that leads into the atrium, and the bulboventricular loop, the ventricular part of it, and then the bulbus conus leading into the truncus arteriosus that splits off into the first pharyngeal arches. You see that there is a remnant of the dorsal mesocardium, and then the transverse pericardial sinus is beginning to appear dorsally there as the dorsal mesocardium is disappearing. So the heart, as we know, becomes suspended within the pericardial cavity, and we see the epicardium has thickened, occupied now the space where the cardiac jelly was, and the endocardium, the endothelial lining is present, and the blood is flowing through the chamber. So the major parts of the heart in the prenatal stage then, truncus arteriosus, it's that tube that is continuous cranially with the aortic sac, and it becomes the pulmonary trunk in the ascending aorta. The bulbus cordus that I had mentioned, I called it conus, bulbus cordus, becomes the inferior part of the aorta and part of the right and the left ventricles. Then there is the primitive ventricle that we saw, the primitive atrium, and that sinus venosus that receives the umbilical veins. The vitelline and the common cardinal veins also drain into this, and it gives rise to the coronary sinus and part of the right atrium. And you'll see all that detail in lecture 28. What are the derivatives of the left and the right horn of the sinus venosus? The sinus venosus has two horns, and again, that will be shown in detail in lecture 28. So the left one is going to become the oblique vein of the left atrium of the heart and part of the coronary sinus. So the left portion of this venous return structure is going to become dedicated to draining the blood from the heart into the right atrium. So the right horn of the sinus venosus is going to become incorporated into the right atrium, and there's just a little part of the right atrium that has a nice smooth wall on it where on the posterior part of it, where the inferior and superior vena cavas drain into it, and that is what the right horn of the sinus venosus becomes. So we all know that there's an atrium on the heart, and the inner lining of, an auricle on the atria of the heart, and the inner wall of the auricles is roughened. There's some elevated structures in that area. Some of the myocardial bands are in that area. So the smooth portion of the wall is what's formed by the sinus venosus. So here is an illustration. These are two simultaneous morphogenic processes that we're going to discuss in this illustration that follow the endocardial tube fusion and produce the adult heart structure. It's septation and looping. So we said that there's kind of a bending or twisting. That's the looping, basically. And then the septation, we said that the myocardial wall is thickening in areas and forms bulges or cushions that are going to meet with each other. So we see the true appearance in the lower left panel. We see the numbers 1, 2, 3, 4, 5 going from caudal to rostral. So now we know it's venous end. Venous end to arterial end. So number 1 is sinus venosus. When we stretch this out and take a look at it, 1 is sinus venosus. I was just looking, there's an error on this. They do. Yeah, this is wrong. So your task is to correct this. So 1 is on the venous end. This should be, good God, what did they do? If this is rostral, then it must be the arterial end. So this should be the truncus arteriosus. This should be the bulbus cordis. This should be the ventricle. And then it should be the atrium and the sinus venosus. So not the greatest thing to have here, huh? But we can see if we look in this one, we recognize it from the illustrations we just saw. So the sinus venosus opens into the primordial atrium, and then there's the ventricle, the bulbus cordis, and then the truncus arteriosus. So in your notes, you need to correct these lines. I will tell you now, this illustration shows up on the exam, so you are challenged to correct the error in this illustration. So the blood flow at this time is an ebb and flow, so it's just kind of a sloshing back and forth with an overall bulk flow from venous through arterial. And we want to name the parts of the heart tube, which can be identified after the formation of the cardiac tube. So the bulbus cordis, the ventricle, the atrium, and the sinus venosum. So these are the structures that should be you should be able to identify on the previous illustration. That went actually quite fast. That completes the lecture for today. I won't go into the lecture 28 because I've already recorded 28A, the development of the venous system, and 28B. I guess we had a quick short class today. I appreciate your time and attention. I'll be here for a while if you have any questions.
Dr. Felix?
Yes.
Dr. Felix?
Yes. Can you hear me?
Yes. I had a couple questions on the test. Do you want us to go over those now, or do you want to do that over email? Or
Just signing on to WebCT. Fire away.
Okay. Well, first on 17. Well, I had two questions over the pictures, and then 1 or 2 questions over the other questions. But on number 17, the question was, let me look real fast.
I'm opening the slides for that portion right now.
Okay, it was the germ cell layer of origin of the central vein. And on the test, when I watched that lecture, you said that hold on.
Waiting for mine to.
I believe that you said that the bile duct and was endodermal origin, and that the rest of it was all mesenchymal origin. And we were kind of confused when we were studying. I remember that I asked you, we were trying to differentiate between mesoderm and mesenchyme, and obviously in a lot of places mesenchyme comes from the neural crest cells, but on the test, I selected D, mesenchyme, but
Now, is mesenchyme a germ cell layer?
No, it's not. But mesoderm, and I know this is sort of picky or whatever, but it was misspelled on the test, MOSODERM, and
that was not intentional.
Right. And I realize that now that I missed it. But that's why I selected the wrong answer. But then then another thing is on number 4, it was germ cell layer of origin, and on that one but that's mesoderm, splanchnic mesoderm.
Still mesoderm, but the best possible answer was the splanchnic mesoderm, right?
I guess. So
now keep in mind that what you're explaining right now, you can submit in writing for reconsideration, anytime during the rest of the semester to get points. So for obviously for the one where it was misspelled, we can give you the full credit on that. On this one, you can get a partial credit because you were correct, but not selecting the best possible answer as we had determined.
Okay, so but just for clarification, splanchnic mesoderm is a germ cell layer, because in the notes, a lot of places it was calling it splanchnic mesenchyme, and I still don't have a full grasp of the difference there. And trying to read in the book, I really couldn't pick anything up.
At this early stage, where we're looking at, say, the 3-week, 4-week embryo, and the mesodermal cells escaped the embryo, so there was the intraembryonic mesoderm and the extraembryonic mesoderm, and that went around the yolk sac was the extraembryonic mesoderm, splanchnic mesoderm, and that went around the the amniotic sac was the somatic, extraembryonic somatic mesoderm. So after a while, 4th week, 5th week, those mesodermal cells are moving forward in their differentiation, and they just are called mesenchyme. And I think it has more, or the origin of the terminology has more to do with the appearance of the cells, and most likely the recent gain in knowledge of the molecular events contributes to it. But histologically, the mesodermal cells are looking like migratory cells, and then the mesenchymal cells are looking a little bit more like fixed cells that are ready to stay in that place and begin to differentiate into things. So yes, it is just like walking along the shoreline, and you're how far into the ocean are you, ankle deep or knee deep? You're still in the ocean. That's not the greatest metaphor, I know, but and it, just lets me play a few tricks on the exam that probably are not nice.
Well, all right. Well, thanks. really just trying to get clarification for the most part, but that helps a lot.
But obviously, there's only the 3 germ cell layers: ectoderm, mesoderm, and endoderm. So
right.
That makes it very specific. And I agree with you. Tagging on the splanchnic muddies the water for mesoderm versus mesenchyme. But I appreciate your tolerance for that.
Okay. On number 18, it's the next slide. It says identify the mesenteric structure at the tip of the arrow. When I watched the lecture on this, I think it said that structure was the gastroduodenal ligament, part of the lesser omentum. And on the test, the correct answer appeared to be the hepatoduodenal ligament, not the gastroduodenal ligament. And when you were going over the question in class on Monday, you kind of drew the whole thing. So you were kind of...
I kind of wandered over. If you notice, I kind of wandered over and was pointing out those 3 things that when I realized, oh, crap. So we'll give everybody that one. But I tried to work with it and realized I was in a mess there.
Well, is the correct answer supposed to be hepatoduodenal ligament?
Well, the way I messed around on the slide, it could be both, right? Because I said, well, this one here, and then I'm looking, that's the stomach and that's the duodenum, right? So that should be gastroduodenal ligament. And I just kind of wandered over and said, you see these things in here? Yeah, so, yeah, guilty as charged.
Okay. And
then someone had brought to my attention number 36. I believe I put mesothelium as the correct response. Did that question give anyone in the classroom some trouble?
Yes, sir. I missed that one also, as mesothelium being the correct response.
You selected
no,
mesothelium?
I selected mesothelium, and the correct response was that you had, or that was put in was dorsal mesoduodenum.
Oh yeah, because that disappears when the duodenum becomes secondarily retroperitoneal. Yeah, that was my reasoning when I chose it for the key.
Oh, so that is the correct response?
Dorsal mesoduodenum. Because all of the mesentery does have mesothelium on it. It has that thin layer of cells, the simple squamous epithelium, called mesothelium.
Okay.
Again, you can argue for points on any question. You have until basically the last day of finals week to turn in any kinds of requests, including reminders of promises I might have made on exam 1 and still haven't come through with yet.
I guess that's all that I have, Dr. Felix. Thanks for
Okay, thanks. I'll still be here for a little while just in case.
You put C and it was wrong. Dr. Felix?
Yes.
One last question. On number 35, define dorsal mesentery. It doesn't look like there was a correct answer selected, just because on all the other ones that there's 100% beside the correct one. But I chose A, and I figure A is wrong after I took the test. When I look back, it is a double layer of peritoneum, but it's not a neurovascular bundle from the post. It encompasses the gastrointestinal tract instead of carries a neurovascular bundle to it, I guess.
A should be the correct response on that.
A should?
Yeah, it's
Okay.
got the 2 layers, and then we've always said neurovascular bundle, and then we throw in the lymphatics, right?
Right.
And it extends from posteriorly, where the dorsal aorta is, to the gastrointestinal tract.
Okay. Well, I selected A, and it was incorrect.
Okay, I'll check the key on that.
Okay. Huh? That's number 35.
Dr. Felix?
Yes.
That one, number 35, doesn't have a correct answer selected. So,
You in there looking at the edit right now?
yes, sir. I just changed it to have A be the correct answer.
Okay, sounds good. Thanks.
Yes, sir.
I was just about to do that myself.
Okay, I got it.
Thank you.
I don't know.
17, he said that mesoderm is the correct answer. He accidentally misspelled it. Okay. And then on 18, I bet he said anything is right on 18, except for the
18, that's a given. At some point, it might be offline in Microsoft Excel spreadsheet. We'll give everyone the 1 point, or it'll be 2 points, 2% points on that one.
Dr. Felix?
Yes.
What did you say we're doing about number 18?
We'll have to give everyone credit for that one.
So, any answer is correct?
Yeah.
Or are there some that aren't?
Well, I think there's some selections that could be wrong, but
So you just, I mean, as far as grading.
it is possible to go in and have more than one correct answer, right?
Yes. So do I select all 4?
liver has to be on.
You're seeing the responses there?
Yes, sir.
State the question again, please.
The question is in reference to slide 11.
It can be either the gastroduodenal or hepatoduodenal ligaments.
Not the lesser omentum, and not the liver. Is that correct?
Correct.
Okay.
Let me know when you've changed that, and I'll regrade again. So how do you have to set it up? Giving questions equal weight or answers equal weight, or what?
Yes, sir. Either answer is 100%.
Okay.
Is that what you wanted? I can do it differently.
Oh, that's the way I want it, yes.
Okay.
Because when I fixed the 35 or whatever it was
Okay.
and regraded, one person went up to 100%. So
Okay.
not given equal weight, they'll end up with less than 100. 99, I guess.
Yes, I just did it.
All set?
Yes, sir.
Okay. And we are regraded. Gotta love this system. Even more and more as I get to know it better.
Yep.
Thanks, Dr. Felix.
Bye. Well, if there are no more questions, I'm going to head off to my meeting.
Have a good one, sir.
Thanks. Bye-bye. See you in a week.